Good morning, students. I welcome you for the biology class. In the previous class, you have studied about the transport in cell membrane, active, passive, diffusion, osmosis in hypertonic, isotonic, and hypotonic solutions, and about mediated transport and all. Today, in this video, we'll learn about nucleus, cytoplasm, and organelles present in cytoplasm. So we'll study only about endoplasmic reticulum in this video, and other organelles we'll be studying in the next video. First, let us study about nucleus. Nucleus is very dense, spherical, largest, prominent organelle which is present in eukaryotes. The nucleus typically consisting of following components. They are nuclear envelope. Nucleus is bounded by two membranes, both forming a nuclear envelope. This nuclear envelope have many pores, which is known as nuclear pore. In between, you do have some space. There's an outer membrane and inner membrane. Inside there's a space. In between, you have nuclear pore. Then, nuclear sap or nucleoplasm. The fluid which is present inside the nucleus is called nucleoplasm. Chromatin material. Chromatin, it's a thread-like material present in a cell. The chromatin organizes itself into chromosome whenever the cell is about to divide. Nucleus contains chromosome and chromosome in turn contains genes which are the centers for genetic information. Nucleolus. Nucleolus is a dense, rounded bodies which is rich in RNAs and protein. Nuclear matrix. The protein containing fibrils joins to nuclear envelope which maintains the shape of the nucleus. So this is a structure. This is a nucleolus, nuclear pore, nuclear plasm. This is a nuclear membrane. This is a two-fold nuclear membrane. They have outer membrane and inner membrane. In between you have a space. And this thread-like structure, it's chromatin. But the chromosome contains two types of things, that is DNA and protein. This is responsible, DNA is responsible for organizing and constructing a new cell. Protein helps in packing and condensation of DNA. So this is chromatin. You can see like noodles, right? So these are nuclear pore. And this chromatin, so it's like thin elongated strands and you can see histones. So these are proteins. So this you'll be studying in your higher classes right now only know. These are made up of histone. The protein content is also there and nucleosomes are also there. So there is a proteins and on which you can see those chromatins are entangled, right? And when you just separate those things, you can get DNA helix, hmm, DNA strands, right? First, let us know what are chromosome. During the growth phase of the cell, the chromatin condenses into much thicker structure called chromosomes. So what are chromatin? That's a thread-like structure which serves as genetic material which is present inside the nucleus of the cell which is made up of DNA and protein. The DNA contains the hereditary information needed for the structure and function of an organism. So this is nucleus and this we have already studied in prokaryotes. They doesn't have nucleus. What they have? A region where chromosome is present is known as nucleoid. So let us study the difference between nucleus and nucleoid, eukaryotic and prokaryotic. So nucleus is a membrane-bound structure in which eukaryotic stores their genetic material and it is well organized and it is larger. It is surrounded by a double membrane called nuclear membrane or envelope which separates nucleus from other cell organelles or you can say cytoplasm. Nucleus contains many chromosomes. It's spherically shaped organelle. Nucleoplasm and nucleolus are present in nucleus. It is composed of DNA, RNA, enzymes, dissolved ions, histones, and other subnuclear bodies. Coming to nucleoid. Nucleoid is a particular area in which prokaryotes store their genetic material. It is poorly organized and it is smaller. It lacks a protective membrane. It doesn't have membrane. It usually contains only one chromosome and it is irregularly shaped organelle. Nucleoplasm, nucleolus are absent. It is composed of DNA, RNA, histones, and other proteins. Now, let us study about cytoplasm. Cytoplasm, it's a jelly-like, viscous, colorless, semi-fluid substance which occurs between plasma membrane and nuclear membrane. The aqueous ground substance of cytoplasm is called cytosol that contains a variety of cell organelles and other insoluble waste products, storage products like starch, glycogen, lipid, etc. In the previous videos, we have studied about protoplasm, protoplast, cytoplasm, cytosol. So please do recollect. And what is the function case of cytoplasm? It's a site of certain metabolic reactions. So many metabolic reactions happens in cytoplasm as it contains many stuff like amino acids, starch, glycogen. So like glycolysis, synthesis of lipids, the fatty acids, nucleotides happen in cytoplasm. What is the significance of membrane bound organelles? So in the cytoplasm there is organelles, right? So cells perform several functions. These organelles are useful to carry all those separate functions. The organelles are endoplasmic reticulum, Golgi, lysosome, mitochondria, plastids, vacuoles, centrioles, ribosomes, peroxisomes. So we have to learn all these organelles. So here's a image for you where all these organelles are present, or you can go through your previous class notes. In each standard also I've written all this structure. First, in this video, let us study only the one particular organelle, that is endoplasmic reticulum. So endoplasmic reticulum, you can see the structure here. It is an extension from the nuclear membrane. There's a nuclear membrane thing, extension from the nuclear membrane. Okay, so what are the things labelings are there? Cristae is there, ribosome, rough endoplasmic reticulum, and a smooth endoplasmic reticulum. There is cristernal space also in between. So let us learn about the structure. ER. It's an extensive network of intracellular membrane which is bound in the form of tubules as well as vesicles that occupies most of the cytoplasm. It looks like very long tubes, or it can be round, or it can be in the form of vesicles, right? So it extends from nucleus. It is attached to the nuclear membrane, and from there it extends till the plasma membrane. So it has a very much have an extensive network. Two types of ER are rough endoplasmic reticulum and smooth endoplasmic reticulum. So before that, let us learn, like whenever it is extending, you can see a tubule sort of a thing. So it is known as cristae. So usually rough endoplasmic reticulum has in the form of cristae, folded structures. Whereas in the form of tubes or vesicles, it will be smooth endoplasmic reticulum. So rough will be towards the nuclear membrane and smooth, you can say, it is away from the nuclear membrane. Coming to rough endoplasmic reticulum, it is also called granular because they do have ribosomes. So rough, why it is called because rough? Because ribosomes are attached to it. So this ribosomes is also made up of nucleic acids and proteins. So this is a site of protein synthesis. The rough endoplasmic reticulum is also involved in the modifications and folding of protein. So proper structure of protein is given by rough endoplasmic reticulum. Coming to smooth ER or agranular, as the name itself indicates, they doesn't have ribosome. Smooth endoplasmic reticulum do not have ribosome and they are not involved in protein synthesis. However, they are involved in synthesis of lipids and other substances. So mainly it helps to detoxifying the poisonous substances. Now, difference between RER and SER. RER, it's in the form of cristae, it is towards the nucleus. and tubules and vesicle that form you can see in case of smooth endoplasmic reticulum. In RER, ribosomes is attached, whereas in SER it is free of ribosome. RER, where ribosomes are present, protein synthesis takes place, whereas in SER, synthesis of steroid hormones, lipids, cholesterol takes place in SER. And RER, the products which are produced are passed into the lumen of ER. So in between those spaces you have crystalline spaces and all. So there's a lumen where this products will move and get modified. So proteins gets modified by means of transport vesicles and then it buds off from the cristae and then it moves to Golgi for further modification. Whereas in SER, products do not pass into lumen. Where RER, it is most abundant in exocrine pancreatic cells where more and more proteins are required. Whereas SCR is abundant in liver cells where more lipid synthesis is required. Coming to functions of ER. It has an extensive network, so it brings about support to the cells. And it helps in transportation of materials from cell membrane to nucleus or from cytoplasm to nucleus. And there's an exchange of materials can also take place from nucleus to cytoplasm or from cytoplasm to cell membrane. So materials gets moved by this ER. And localization of organelles. So organelles are stationized to their respective position by this extensive network of ER. Formation of organelles like Golgi, lysosome, vacuole, it's from SCR. Surface for protein synthesis, whereas RER is required for protein synthesis and SER is required for lipid synthesis. And packaging. So ER is required for packaging in the sense the protein which are formed from RER, it moves into the lumen and gets packed as a transport material. It butts off from the cristae and then it starts moving to Golgi. So in Golgi, further modification of proteins, a proper structure, addition of some other materials takes place and that's how proteins are formed. Detoxification. SCR is involved in detoxification where the poisonous substances, pollutants, drugs which is present in our body is detoxified and it is excreted out by means of SCR. And many like proteins, lipids, cholesterol, steroids synthesis is happening in ER and those proteins and lipids which is synthesized it's utilized for the cell membrane itself. No? The cell membrane itself uses some of these materials to synthesize. This is called membrane biogenesis. It's important. The proteins and lipids which are synthesized in the cells with the help of ER are used to build the cell membrane. This is an assignment for you. Draw a neat label diagram of endoplasmic reticulum. Thank you. Let us study about ribosomes and other organelles in the next class. Until then, thank you. Have a nice day.